So cell signaling or signal transduction is going to get a functional signal from outside the cell, inside the cell. And so we're going to do a lot of sensing, okay? Whether it be consciously, so sending a signal from our thoughts and processes about doing something, or autonomically, where we are detecting things we don't even know. But our body and our cell types are going to be responding in different ways to different things. And so we're going to talk about the very many ways in which a signal from outside the cell gets inside the cell and starts up a process inside the cell. The signal transduction cascade has the ability to sense, process, and respond to all sorts of stimuli, whether we know it or not. It employs receptors, enzymes, channels, regulatory proteins, and they will work in concert in what we call molecular circuit. Okay? Another way to think about it is kind of like a domino effect. One triggers the next, triggers the next, triggers the next, until we finally get to the end of our process. And the molecular circuits have a couple of characteristics. They can detect, they can amplify, they can integrate the signals. So detect, amplify, and integrate. Now, features of a signal transduction is some sort of specificity. We're going to amplify. We're going to see that there is desensitization. We're going to be desensitized or adapt to a signal, and then we're going to have integration. All right. What in the world do you think this one represents of those? It's just like a test question. You got 4 choices. What are you going to go? Desensitize. We're going to desensitize. All right, here's something that's really oddball. Every time we send a signal, almost always, there's a turn-off mechanism that starts immediately. Okay? Here in biological systems, we never want to start something on and never turn it off. And so we almost have an auto-off sequence with anything that we see. Okay? What do you think this one represents? Amplification. One signal molecule can produce hundreds of thousands of responses on the inside of a cell as part of the amplification. Up here, sensitivity. We want to be able to detect or cause our tissue types to respond in certain ways, and so you may either respond to insulin or glucagon, both of which are signaling for different levels of glucose in your bloodstream, and your body can tell the difference between the two signals because they are opposite. And then what in the world? Well, by elimination, this one is what? It's that integration. And so what happens is there are some tissue types and some enzymes that get multiple signals. Sometimes they get a signal to increase, and sometimes they get even a signal maybe to decrease. And what the body is fine-tuning is not an on-off switch, but I want it to run at 60%. So I upregulate it 100% on one side, and you downregulate it by 40% on the other, and you get 60% net. Okay? A lot of times it really is interplay between two different pathways that intersect in the same place, and so you may not need to be 100% active to fund everything, but it becomes complex. So examples of general types of signal transducers. We will see an example of each. Our first, simply an ion channel. What we're going to do is we're going to send a signal across the membrane, and that signal will be a change in ion concentration. Okay? So we can open up a channel specifically, and what comes through are ions. And when that happens, I have a change inside the cell for a process. All right, a lot more often, Okay, we can have a receptor enzyme. Okay, receptor enzyme. On one side, on the outside, the molecule binds to a signal. It receives, is the receptor of the signal. When that happens, small conformational changes occur throughout the entire protein. On the inside of this cell, an enzyme without binding to a molecule is inactive, but when a molecule binds and all of those conformational changes occur, I change the shape, I become active, the enzyme is functional, that can now send a process inside. Okay, here is a very similar process, serpentine receptor, where the signal will bind a receptor. The receptor doesn't actually, it's not an enzyme, but what it'll do is it will upregulate other proteins to go down the chain and upregulate an enzyme to function. Steroids. Steroid receptors actually occur inside, most of the time, the nucleus themselves, because the steroids are hydrophobic, can get through the membrane. The receptor itself that is inside the nucleus is usually a transcription factor, which is inactive when there's no steroid present and active when there is steroid present. The ultimate signal here is to upregulate the DNA to produce a protein or a gene product. Okay. Then we have receptors that will bind and cause coordination of proteins to come together, which causes an event. And then lastly, there is some, and they put in this class, we won't see an example of this in this case, but some adhesion molecules, proteins that hold other proteins together, cause signaling between the proteins, and those are small conformational changes. So we'll get examples of everything that I've boxed around. In general, what we're going to see is some sort of a release on the outside of cells, and usually it will be small molecules. It can be as small as an organic molecule or a small protein. The small protein will bind to a receptor. That receptor, in some way, shape, or form, the binding on the outside causes something on the inside to happen. In this case, phosphorylation on the inside, which then creates binding pockets for molecules that weren't existing before. And once they bind, then they can go out and they can tell friends about it. And that's just like you hear something juicy and you just tell one person, right? Text one person. And by the time you get across campus, it's everywhere. All right. So, first example, ion channels. Ion channels will interface, and what we're going to talk about is a nerve impulse. Okay? So, the channels can be controlled in terms of when they open or when they close in two different ways. One is called ligand-gated, and one is called voltage-gated. A ligand is this wonderful, generic, scientific term that means thing. Okay? Thing. It's going to bind something. Okay? The thing is the ligand. Okay? ATP can be the ligand. Whatever can be the ligand. It's another word, another good scientific word you can use for thing is moiety. It's a thing that binds. Okay? And so we're going to look at the specific one, the acetylcholine receptor. The ligand, the thing, will be acetylcholine. Voltage-gated means that the channel will open in response to a voltage change across the membrane. Now, voltage change means there's an electrical change, right? Is there electricity moving through us? It's what? It's charges moving around. Now, actually, at a most basic level, it could even look a little bit like electricity, but really we're talking about ion changes moving across. We'll look at the sodium, potassium, and calcium channels as examples of those. Okay, so the synapse. This is where these molecules are these signal transducers are found most often. All right, we have a neuron, and on the other side we have muscle, or you could go to another neuron, but I'm sending a signal between the neuron and its next stage. The neurotransmitter is in vesicles inside right at the end of the neuron. The neurotransmitter, in this case specifically, is the molecule acetylcholine. I'll show you what that structure looks like in just a moment. Okay. The acetylcholine inside the vesicle, not a signal molecule. The acetylcholine that is released in the cleft is the signaling molecule. All right, and what's going to happen is we're going to have a nerve impulse come down. The vesicle is going to fuse to the membrane, and this neurotransmitter is going to be released. Okay? That neurotransmitter then sends the signal from one to the next. Yes?
Is that what happens when we travel across neurons too?
It goes for, that's the same thing, neuron to neuron, or neuron to the muscle itself. And so the arrival of a nerve impulse, which is a voltage change, comes down the pike. Acetylcholine is released into the cleft between either two nerve cells or between the nerve cell and the muscle. This acetylcholine, the acetylcholine is released, and the cleft is between the two nerve cells, not too wide, about 50 nanometers. And it makes a very large concentration in that little cleft for just a short period of time. Here is that acetylcholine. All right. In terms of, it's made up of an acetyl group. Okay. Something that you probably should know. And something that you might not know, choline, which is an alcohol, where originally it was an alcohol. 2 carbons with a nitrogen, 2 carbons with a nitrogen that has methyl groups on it. We'll see choline quite a lot. Okay, so here's acetylcholine. The receptor is called the acetylcholine receptor or the nicotinic acetylcholine receptor, and you'll see both of those. When the nerve impulse reaches the end of a nerve, it releases about 3 times 10 to the 6 molecules of acetylcholine. What is, give me a real number of that. What is 3 times 10 to the 6th? 3 million. So 3 million of these things are released. Okay. And they can diffuse in that cleft. There are 2 places in which the acetylcholine can bind. First, it can bind to the receptor. This is what causes the next step on the presynaptic or the postsynaptic membrane or on the muscle cell. Or 2, it can diffuse into the active site of an enzyme called acetylcholinesterase. What do you think acetylcholinesterase will do? Break it down. Okay. And if you go back just a second, what is this functional group? An ester. Okay. When the acetylcholine binds the receptor, it opens up a channel. All right, and ions flow. Okay, when you take a look at that, we see sodium goes in, potassium goes out. From that picture, what else do you see about the sodium and potassium out? Which one's bigger? Sodium's influx is larger than potassium's efflux. Why is that? Why does more sodium move in, less potassium move out? What is it?
Because the sodium potassium pumps have been.
Yes, the sodium potassium pump has created a gradient, right? But is it even? No. There's a bigger difference in sodium, smaller difference in potassium. So when you open up a channel to allow them to move, just the fact that there's bigger gradient for sodium, more sodium moves through. Okay? Now, let's think about that. The sodium, potassium, ATPase, which you've already learned about, left what kind of charge on the inside of the cell, on the outside of the cell? Once the pump has finished, what is the charge difference? Inside is what? Not positive. So inside is negative, outside is positive. Why is that? Remember, 3 out, 3 positives out, 2 positives in, right? And so more positives on the outside, less on the inside, so that makes it relatively negative. When the channel opens up here, what happens? Lots of sodiums rush in, not as many potassiums rush in. The cell, the nerve cell, was once negative on the inside, positive on the outside. When the acetylcholine receptor is opened up, it's called depolarizes, or it makes it more negative on the outside and more positive on the inside. It changes its normal state. Was negative on the inside after the acetylcholine is released. It is now positive right around where those receptors are. So in the nerve, it depolarizes, but it depolarizes kind of right here. Okay? Way down here, way down here it's still negative. But right up here, it's changed. Because remember, if you actually, let's say you take a sugar cube and you drop it into water and it just starts to melt, you can see how it diffuses. Well, that's kind of how it happens. And so here it becomes positive. Here it's still negative. And so to continue this whole process, I've got to get the rest of this changed. My receptors on this side doesn't change the entire length, especially a long nerve. Okay? So we've got some other things that have to happen. The action potential is that change from negative to positive. That's what the action potential is called. Consequence 2. An enzyme. Okay? And then I write CHEM-321. What in the world does that mean? It's a serine. So I have a big old protein that may have 350 amino acids, but one's important, the serine. Okay? And it's going to break an ester bond. So the acetylcholine will bind into the enzyme, and there's the acetylcholine, the acetyl group with the choline. Okay? And what happens is this hydroxide will attack that ketone. Okay? I feel like doing some chemistry. Okay, take that proton. This will attack the carbonyl. I will kick up. I will kick down. And this will pick up a proton. And I have now just broken the acetylcholine. Choline falls off. The acetyl group is now on the serine. Okay. Then the regeneration step, water has to come in. It will attack the carbonyl also, kick up, kick down, and the acetate will fall off. Neither choline nor acetate will signal, but acetylcholine does fit into the enzyme. What does this mechanism resemble? What enzyme does this acetylcholine esterase resemble? And a mechanism that you have learned or were introduced to. Let's see.
Is it trypsin?
Which one?
Trypsin?
Trypsin, amitripsin, and elastase. Very similar in mechanism, where when we had the peptide for the protease, serine protease, it came in and attacked, part of it fell off, part of it was stuck on, water had to come in and react a second time to get the other piece off. So, very similar in action to the serine proteases in terms of mechanisms. Okay? The signal for the acetylcholine receptor will run as long as there is acetylcholine in that cleft. And so the esterase, how fast it processes, allows us to reset the nerve so it can send another signal. And so 3 million are in that cleft. The acetylcholine esterase has to come back and cleave 3 million of them to stop the signal. All right, so that's the signaling molecule. The acetylcholine receptor itself is a pretty big protein. Okay, it has 5 subunits, 2 alphas, a beta, a gamma, and a delta. Okay, because of the sequence of these 5 different subunits, when I look at them, the sequences of these genes are almost identical. There are only small differences between the alpha, beta, gamma, and delta. The idea about this is that the subunits must have come together out of a divergent process in which there was one gene, and then it got copied, it got a little changed, worked a little better. Another gene got copied, it changed, got a little better, until we got all 5. So divergence from a common ancestral gene is the idea. That's how the book describes that. There are 2 acetylcholine binding sites, and if you have 2 binding sites and there are 2 alpha subunits, that was a pretty good idea where they were, and we did find out that they were 1 each in the alpha subunits. Here is a very detailed structure of them, and if you think about what this is, okay, it is a receptor, okay, that becomes a channel. Important pieces of a channel. You've got to be able to go through the membrane. So here is a place that goes through the membrane. On this side, I just have to open up and dump ions through. On this side, this is where the acetylcholine is going to be bound. So this has to receive the signal and cause a change so that this opening opens enough for ions to flow. What's interesting about this is that these 5 come together and you get a primary 1 alpha helix that come together right at the interface between all 5 that create that channel that's big enough for the ions to flow. So rather large on the synaptic side, rather small on the cytosolic side, but here's where all the detection happens, so this is where all the protein is. All right. What I'm looking at and I'm focusing in on are these alpha helix, okay, the 5. There's an alpha helix, there's an alpha helix. I get really technical there, right? Okay. Alpha helix, alpha helix, alpha helix, alpha helix, alpha helix. And what in the world is this? It's an L. It means it's, no, that's a good guess. It's not the ligand. It is a large side chain, okay? Takes up a lot of space. Large side chain. And so the hole is there, but if I have side chains in the middle, that means the hole is closed. In the unstimulated, or when there is no acetylcholine bound, the alpha helix have large subunits that just physically block the hole. Okay. I have an L there. I also have an S there. What does that S mean? Small. Oh, we're cooking with gas now. When the acetylcholine bind, the small conformational change makes each alpha helix rotate just a little bit, moving the large out of the way, putting the small in the center. Now I have 5 smalls. I got me a hole. I've got a hole, things can flow through. And that's essentially what this does. Now wait a minute. What did I say flows through the acetylcholine receptor? What?
Said ions.
Ions. Which ones?
Acetylcholine.
Not acetylcholine.
Or,
Sodium and potassium. Well, if I create a hole, shouldn't everything flow?
Just big enough for the...
Okay, one has to be just big enough. Nothing too big can get in there, so it has to be at least... Ah. I didn't mention anything about anything negative coming through, right? And so there has to be some sort of selectivity. Okay? Here is what happens. In those alpha helix, not only is it a large and a small, but check out the side chains that are ringing those alpha helix. They're all what? Negative, right? Am I going to have a negative ion go through that channel? No. The negative will repulse negatives. So even if there is a gradient of negatives, they just can't get through this particular channel. They will go through other channels, but not this one. Okay? I can allow sodium and potassium to flow through. Are sodium and potassium the only ions that are positive? So why are those the ones that primarily move? There's a lot of them. There's a gradient of them. All of the others around most cells have a pretty even concentration on either side. So if you open up a hole and there's no concentration difference, there's relatively no movement. Okay? And the only reason why we have this big sodium-potassium is because of the sodium-potassium ATPase, or that pump, that is active in almost all cells. Okay? Now, as you have the nerve impulse, I told you, the acetylcholine receptor just changes the concentration, makes it positive right along the edge of the top. But a nerve cell is very long, and it's still negative all the way down. Why did I get negative all the way down? Well, what happens is, on that receptor, okay, if I look at the cell like this, I'm a wonderful artist, let me tell you. Okay? And I made that right there. Can you even see a difference in color there? Okay, I can't, so that's good for you. All right. Right here is where we need to start working on it. That still needs to change. Okay? What happens is I have channels along the edges. I have sodium and potassium channels along the edges. Okay? And these sodium and potassium channels are signaled when there is a change in the gradient. And so as this diffuses down in this area, what's going to happen is that channel and this channel start to see a change in the ion concentration. That will cause them to open, allowing influx and efflux, which then causes that area to diffuse down, which causes other ones to open, which causes other ones to open, which causes other ones to open, as we get through the entire sequence. Okay? And so we're allowing all of these things to open and go through and depolarize. Here's the problem. That nerve cell, once you've depolarized it, is dead. Until what? You have to repolarize it. How do you repolarize it? You have to close all the channels, and then you have to have the sodium potassium ATPase pump out. Okay? And so, well, we learned that pump. It's going to be working here in the membranes too, fixing or resetting all of these. Okay. The sodium channel. Here is its protein. Okay, it's a single polypeptide chain, 4 repeating subunits, 1 through 4. Each kind of repeating unit has 6 transmembrane helices, and in each of those, they're numbered 1 through 6. The 4th alpha helix is the important part. It's called the voltage center. Now, if you take a look at that protein, it's got 24 transmembrane helices. This thing is huge, okay? But it's rather important. And it's a very large protein, embedded very much in the membrane. The S4 sensor will twist when there's a change in voltage that it senses. And as it does, it breaks bonds, and that's what causes the conformational change to change the whole structure from closed to open. And so we know this particular alpha helix is the one that detects that change. I said, when it suddenly changes, it'll open up. That's where it does. And it does by rotating the alpha helix. The sodium channel itself, the pore size, is about 5 angstroms large, and it is negatively charged or aligned. Okay? It allows passage of one sodium and one water complex together. Okay? And so you see that the sodium here along with one water. Okay? Here's a carboxy group of one of the side chains that helps stabilize that positive charge as it comes through. Before, it's actually surrounded by water to make sure that that positive charge is balanced. But when it gets inside the protein, one water comes in and keeps it balanced on one side, but then it substitutes a water for a acid interaction. All right, and then there's an oxygen that's allowed to hydrogen bond to the water as it passes through. So it gets through in this particular sequence. So it is optimized for this 5. Potassium can't get through because why? It's too big. That's half an answer. Potassium is too big to bring potassium and water together. Okay. Look at the radius of potassium. Is it too big to get through the hole physically? It isn't. So why doesn't the potassium just fall through because it's small enough? And it just lets go of water. It would fit, right? Yes. Yeah, it's about that charge. And okay, so potassium does fit, right? But potassium is just too small. It can fit here, and it can interact the positive on these negative charges, but it's not big enough to interact on that side. Or it can interact up here, but it can't interact right here. But if potassium's over here, it's surrounded by water. All sides are stabilized. And it's on this side, it's surrounded by water. All sides are stabilized. But in the middle, even though it is small enough to fit physically, it is not optimized to have interactions all around it. And that's high energy. Why does sodium and water fit? Because it's optimized to be able to make both interactions on both sides of that. And so it is selective for sodium only because of its size. It will not allow potassium to flow, which is it's different than the acetylcholine receptor, which allows both of them to flow. Yes?
Why would lithium not come through since it could have interaction?
Yes. Lithium, there's a couple things about lithium. Again, it would be too small to make interactions on both sides at the same time. So it can't flow through. It would be able to interact on one or the other, but not both. And there's not a huge lithium gradient, so there would be no movement, no reason to move. Now, sodium channels. Here, in their infinite wisdom, there is a depolarization event. Inside the cell becomes changed. It opens up. It allows sodium to come into the cell. Okay. The sodium channel opens up and is active for about a millisecond. Okay. Nerve impulses are very, very quick. You know this because if you've ever cooked and you put your hand down on a hot stove, how fast does your hand move? Pretty quick. Okay. All right. And so what we really want to do is to have depolarization, but we don't want to depolarize it forever. We want to reset it to send another signal. So we open enough to allow enough ions to flow through so that we have the depolarization, but not overwhelm it. Because if I send more and more and more, I have to spend more ATP sending it back, back, and back. Okay? So it opens up for just a moment in time. There is a mechanism called a ball and chain mechanism that closes the channel. When I see the sensor and every one of these S4 sensor detects here, 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 and over here, they all twist. They all create a pocket. They all create an opening so ions flow. But check this little piece of protein out right here. So the protein is like floating around. In the closed state, it floats around pretty well. But when the open state opens up, oh, check this out. That little thing that floats around fits right as a cork inside the open hole. The length of time it takes to get there is dependent upon how large that tether is. And that tether is 1 millisecond long in terms of its ability. Normally, that piece of protein doesn't interact unless it's open. Once it's open, it will plug up real quickly. It's a desensitization mechanism, or an adapting, so that we can reactivate and repolarize the cell. If I take a look at the potassium channel and the calcium channel, they are very similar to the sodium channel. The potassium and the calcium channel have S4 voltage sensors. They have bundles of 6 alpha helix that work together. The mechanism of action is very, very similar. The difference is both the potassium channels, whether the versions that we find, they are single subunit. So for them to work, they have to work together as a unit of 4 different proteins to come together and work. The calcium channel is even closer to the sodium channel in terms of its function. What's the difference between the calcium and the sodium channel? The pore size itself. They will be optimized for each one of those different sizes that I'm going to allow to flow through. In terms of the potassium channel, the channel pore size is 3 angstroms, which is smaller than the sodium, which is 5. But remember, the sodium allowed sodium and water to come through. The potassium channel is only going to allow potassium to flow through. All right? And so potassium's radius is 1.3 angstroms. Okay? Sodium is just too small to be stabilized on all sides, and so it won't flow through, even though it's smaller. Okay? And we've talked about that. Don't need to talk about that again. Just wanted to let you know that, again, within milliseconds for the potassium channel, it closes. Here's another picture of the ball and chain in activation, which opens up. The one thing that I do want to identify in this particular one is that I said that this piece of protein here is sitting and floating around, and it won't interact with the pocket. When you open up the channel pocket, what really actually opens up is a specificity for it. So this has a positive. There's a positive residue, so it kind of keeps out of the way. But as soon as you have the conformational change, it opens up a negative residue, so now there's an attraction to it. That's why it's attracted there. And so it opens up a channel, allows things to flow through, but then creates a specificity for that ball and chain mechanism to actually function. Okay? And so, as we go down the synapse, sodium in, potassium out, sodium in, potassium out, sodium in, potassium out, sodium in, potassium out. Okay? I did introduce the calcium channel. That's right here. Okay? The calcium channel opens up and will infuse right here near the end of the chain where the vesicles that have the acetylcholine sitting. Okay. The calcium is what's going to cause the vesicles to merge with the membrane and to excitose the acetylcholine into the cleft. Okay? And so I did mention the calcium. Where is it? It's right near the end, near these vesicles. Okay? And then the acetylcholine comes to the receptor opens up, I depolarize, and I continue the action potential down. Okay? All right. What I don't have for you, but I will open up. Okay? I'm going to place an additional worksheet in our up on Moodle. What I'm going to do is I'm going to talk to you about how this calcium works with the neurotransmitters, okay, and messes up the neurotransmitter transmission. Okay, what we're going to do is we're going to read an article about a toxin, a neurotoxin, and how it messes up neurotransmitters. And it's going to affect the calcium interactions. All right? And so it's going to talk about these vesicles, it's going to talk about proteins around the edges and how this all works together. Let me give you a little bit of background to that so that you understand the article a little bit and what I want from it. Okay. Okay, we're going to have the vesicle that has the acetylcholine. Okay. We're going to have the plasma membrane. All right. I've got to get the acetylcholine from inside this vesicle outside into the cleft. Okay. There are going to be some proteins on the vesicle, and there's going to be some proteins on the membrane that have to get together to cause these things to merge. Okay. And what we're going to talk about is the snare complex. The snare complex is soluble in ethyl malamide sensitive factor attachment protein receptor. That's why I call it snare. Okay. There's going to be something called the T snare and the V snare. We're going to use syntaxin, SNAP25, and another protein to actually trap these together. Okay, proteins are going to come together and trap them. All right, we're going to have an interaction between the protein called SNAP25, okay, and syntaxin, okay, with, they're calling it VAMP here, we're going to call it something different in the article, but we're going to have one protein. The proteins have to bind together. If I can get the proteins bound together, I get the vesicle membrane and the plasma membrane close together. Got that? That's the big concept. Okay. The toxin that we're going to use is a botulinum toxin. Okay. Botox. All right. You know what Botox is actually FDA approved for? It is not for wrinkle removal. It is for migraines and excessive sweating. Okay. This is the whole FDA reason why we have Botox. But we have Botox now so that we don't have any wrinkles. Do you know why you don't have any wrinkles? Because what you do is you stop the signals to your muscles. And so if you have no signals to your muscles don't contract. If they don't contract, you don't get wrinkles. Okay? We're going to interrupt the signaling of the cells. Okay? All right. And so, what actually happens is Botox will cut the proteins, and particularly the SNAP25. And there are two different versions, A and E. A cuts a little piece of the C-terminus off, and E cuts a large piece of the C-terminus off. And we're going to look at experiments, what happens when we interact these, what happens with the proteins themselves. Okay? All right, here's the two pieces. These are the three proteins that we're looking at. Synaptotagmin, which has calcium binding spots. Syntaxin and the SNAP25. What you need to understand is the plasma membrane has the Syntaxin and the SNAP25 on this side, and the vesicle has the synaptotachin. Experimentally, to bring these two together, these three have to come together, and it's controlled by what? Calcium. And remember, calcium is only at the end of the nerve cell. So when calcium comes in, that's the signal to bring these things together. All right, what we're going to do is do a lot of experiments with this protein, and that protein in a test tube with or without calcium. And you would think if they get together, they should get together, right? And if it's calcium dependent, they probably should get together better if you put what in there? Calcium. And they're going to do experiments. And then what they're going to do is they're going to add Botox to it and see if those proteins still bind as well together as without Botox. Okay? And so I'll upload the article, I'll upload the activity. It's going to look like a quiz. I don't think I'm going to make it a quiz, but I do want you to work through it. Okay? But it'll show you. It's also, we're going to do the experiment of immunoprecipitation, whereas you mix everything together, you pull down one protein, and if they're actually binding together, all of them should come down. If you pull one protein and nothing comes down, then no protein should actually come. And that's the experiments that we're going to look at. And you're going to learn how Botox works.